Hi guys, Miss McClellan here, coming to you live, giving you your genetics flip notes lesson. Alright, genetics is awesome because the idea is if we can understand how all the genes in an organism work, how they all interact with each other, we can start to create our very own organisms from scratch. We can make our superhumans. We can make our super plants and animals. We can do what we want if we understand all the repercussions of how these genes interact with one another. Well, we've already started to do these things. For example, we've taken the gene that makes poison in the scorpions, and we've taken cabbage, the sweet innocent cabbage, and we've put that gene inside of the cabbage cells. Well, why would you want venomous cabbage, you say? Well, insects, when they bite into the cabbage, die. We've also taken yeast cells that are now producing diesel fuel for us rather than the more common alcohol that they normally make. We've taken goats and spliced them with spider genes so that the goats are actually producing spider silk in their milk. We have trees that clean up pollution in water. We have cats that glow in the dark. And we've got bananas that we're injecting vaccines into their cells so that instead of taking a nasty shot, you can just eat your bananas. Oh, and of course, there's cloning. Now, what's the driving question behind genetics? It's how do we get from a person's genotype, so the exact genes that they have, to their phenotype? The phenotype is just the manifestation of those genes that were given to you by your biological parents. So phenotypes include things like how averse are you to taking risks? How aggressive might you be? Do you have blue eyes or brown eyes? Anything that is who you are as a person, we're wondering how much of that is defined by your genotype. Well, how do we get from genotype to phenotype? As you know, DNA is trapped inside the nucleus of your cells. It has all the information needed for any cell to become any type of cell and for the organism to function as whatever organism it is. Well, how does it do that? The DNA has genes, and the coding strand for the gene will be read and RNA will be created. It's this messenger RNA, and the messenger RNA will take the message from the gene out of the nucleus. And a ribosome in the cytoplasm is going to grab onto that message, and it's going to make a protein from the message. Why? Because proteins are who carry out the instructions that the DNA has. So, genotype is in the DNA. Phenotype, it's expressed by the proteins you have doing work around your cells. All right, well, what are genes made of? It's made out of DNA. Well, what's DNA? Deoxyribonucleic acid. It has 6 molecular components. You have 4 nucleobases: thymine, adenine, cytosine, and guanine. We'll go over these in a little more detail in a second. Each of those nucleobases is then attached to a ribose sugar, and those ribose sugars, these little pentagrams that you see, are then snapped together with phosphates. Okay? So when you flip the page in your guided note sheet, you're going to make a little key at the top of the page. 4 nucleobases, give them a color, say green. Ribose sugar, give it a color. And the phosphates will be yet another color. Okay? So here we go. On the next slide, you see the 4 nucleobases, all right? So take your color for nucleobases and color these guys in. Now, what's holding the 2 sides of the strands together? Right here We see some nitrogens, we see some hydrogens, we see some oxygens, some hydrogens. You've got it, hydrogen bonds. Next, you're going to color code the ribose sugars, which you can see have now been attached to the nucleobases. So take your next color, color in your ribose sugars. Now you can see that you have your phosphates linking the ribose sugars together. So this is what makes the outside of the double helix. So this is one side of the helix is a phosphate with the ribose sugar, phosphate, ribose sugar, phosphate, ribose sugar. Each ribose sugar is attached to a nucleobase. Okay, well, how does that translate to a gene? Well, one side of the double helix is going to be called the coding strand, okay? This is what has the code for the gene in it, the code that will create a messenger RNA and then from that be translated into a protein. So if you have ATTCGCT, that would be the start of, say, a gene. And the order of these nucleobases is what spells out what type of gene you have. Now this could be thousands of bases long, and one error in the order of these bases could lead to a mutation. Now, mutations are most often deleterious, but sometimes they can be to our benefit, helping a species to evolve. The other side of the DNA helix is a complementary strand, and it's simply that DNA is more stable as a double helix than it is as just one strand. So you can see you have the coding strand on one side and then the complementary strand on the other side. Well, what do I mean by complementary strand? How do I know if I have a coding strand, what's going to go on the other side? What always attaches to a T or an A or a C? Well, the cell will always attach a cytosine to a guanine, an adenine to a thymine, a guanine to a cytosine, a thymine to an adenine, always the same. Always the same. Well, how is DNA stored? DNA, if you were to take it and unravel all the DNA inside of the nucleus, stretch it out from end to end, it would be 6 feet long. That's 6 feet of molecule that we have to wrap up into a nucleus inside of a microscopic cell. Okay, if you took all your DNA and stretched end to end from all of your cells, it would go past the sun. That's over 150 million kilometers. All right. Well, how do we package it? So we take the double helix. You'll see on your note sheet that you have a little box pointing to the double helix. And that double helix will then be wrapped around these proteins that are called histones. So these proteins then, once they have the DNA wrapped around themselves, the proteins then wrap further and further, creating this complicated structure called a chromosome. So the second box up from your double helix shows the DNA being wrapped around the proteins, that turning into the chromosome, and then the chromosomes are stored in the nucleus of the cell. Well, each person has 23 pairs of chromosomes. Genes are coded by the DNA on those chromosomes, and specific genes are found on each chromosome. Okay, 23 pairs of chromosomes? Why not just say 46, Miss McClellan? Well, that's because that's ridiculous, okay? It would be like me asking you, how many shoes do you have? You tell me how many pairs of shoes you have. They come in a set, okay? It doesn't matter. You don't say I have 46 shoes. You have 23 pairs. Okay. Why am I saying this? Well, it's because 23 of the chromosomes you got came from your biological mom, and the other 23 of the same exact chromosomes were given to you by your biological dad. So that means there's a little bit of redundancy here, which is a good thing. You have 2 of every chromosome. And you can see they have these special banding patterns on them. Well, this is a karyotype, which is a picture of the chromosomes in somebody's cells. Everybody has 2 of every chromosome. There are some anomalies where people will end up with maybe 3 of a chromosome. And there's usually different phenotypes that result from having a chromosomal difference. If you have 2 of every chromosomes, that also means that you have 2 of every gene. But you might get different versions of the gene, okay? So that's how your phenotypes end up differing. An allele is a different version of a gene. So example, there's a blue eye allele, a brown eye allele. The exact combination of those alleles is your genotype, and then the result of those 2 alleles will be your phenotype, what you end up looking like. Another great thing about having 2 of every gene is if you get a mutated version of a gene that doesn't produce a working protein, sometimes that's okay because usually the other gene that you get isn't also going to be mutated. So it'll be just fine and it will cover your back. All right, guys, thanks so much for listening. We'll have more exciting things on genetics next class I see you, because you'll be learning exactly how to figure out what phenotypes you had, what genotypes you had, and so on and so forth. Thank you so much. Bye-bye.